Good afternoon, everyone. Welcome to the third edition of the National Virtual Leukemia Conference. This is a one-of-a-kind conference that addresses the unique needs of the blood cancer community and offers the latest information, tools, and resources to help Canadians touched by leukemia. The Leukemia and Lymphoma Society of Canada is proud to introduce today's presentation, Understanding RNA and its Role in Leukemia. In this session, Dr. Katherine Borden, who is a professor of pathology and cell biology at the Institute of Research in Immunology and Cancer at the University of Montreal, and Dr. Sarit Assouline is chief of the Division of Hematology at the Jewish General Hospital and senior investigator in the Lady Davis Institute at the Jewish General Hospital. Drs. Borden and Assouline will unpack the scientific wonder that is ribonucleic acid, or RNA. This is the molecule that touches nearly everything in human cells. They will discuss how RNA biology can be disrupted and used as therapy for leukemia and other blood cancers, and why clinical trials are important in this area. Before we begin, let me introduce myself. My name is Paul O'Connell, and I am the Research Program Director at the Leukemia and Lymphoma Society of Canada. I will be the moderator of this online event. We invite you to type your questions and comments in the Q&A box of the Zoom webinar panel throughout the presentation. An LLSC staff member will monitor the questions, and I will read some of your questions aloud at the end of the presentation. Due to the large number of participants and limited time, we may not be able to answer all of your questions. Also note that Drs. Borden and Assouline will not be answering any specific questions about a person's health history or status. Today's presentation will be recorded, therefore you can rewatch it again on our website. Next slide, please. As we come together from across Canada, we recognize that we are on lands cared for by many Indigenous nations. We honor the First Nations, Inuit, and Métis people whose deep connections to these lands enrich our community. We value Indigenous voices, and we are working to ensure they are truly represented in our leadership and staff. At the Leukemia and Lymphoma Society of Canada, our commitment to diversity, inclusion, belonging, and health equity guides how we work to support and uplift everyone affected by blood cancers. Next slide, please. Are you looking for specific programs or resources but not sure where to find them? We are here to help. You can connect with a community services lead in your area to receive personalized assistance and get connected to resources in your area. They can listen attentively and with compassion to the challenges you are facing, point you to information specific to your type of blood cancer or treatment, help you get organized to manage and take charge of your care, and identify service that would be beneficial for you.Reach out to us by calling toll-free at 1-833-222-4884 or visit bloodcancers.ca to find the community services lead in your region. Next slide, please. We know that having a diagnosis of blood cancer can be very overwhelming, but know that you are not alone. There is a blood cancer community that is here to support you every step of the way. As mentioned earlier, you can access one-on-one support to help you navigate the blood cancer experience. Next slide, please. One way we support the blood cancer community is to offer psychosocial and emotional support. We have programs that can help connect you to other members of the blood cancer community. We offer peer support program where you can connect one-on-one with a peer volunteer and virtual support groups. Connect with the community services lead in your area to learn more. Next slide, please. In October, we launched LLSC's Travel to Treatment study. This study is to measure and quantify how traveling for cancer treatment affects individuals and families. We want to gather and present information to decision makers to help them understand the real-life impacts of having to travel for treatment to improve support for individuals affected by blood cancer. This study will examine factors contributing to individual experiences, including finances, education, housing, and employment. To see how travel affects these important life factors, to see how travel affects these important life factors, please fill the survey using the QR code. You'll also receive the direct link to the survey in the follow-up email 24 hours after this session. Thank you for taking a few minutes to fill this important survey. Next slide, please. It is now time to introduce our speakers. Dr. Katherine Borden is a professor of pathology and cell biology and Canada Research Chair in molecular biology. She's an investigator at the Institute for Research in Immunology and Cancer at the University of Montreal and is a full member of the McGill Centre for Translational Research in Cancer. Dr. Sarit Assouline is the Chief of the Division of Hematology at the Jewish General Hospital in Montreal and a Senior Investigator in the Lady Davis Institute at the Jewish General Hospital. She's an Associate Professor in the Department of Medicine and Oncology at McGill University and is also a full member of the McGill Center for Translational Research in Cancer. Thank you very much for being with us here today. Turn it over to you now.
Well, thank you so much, Paul. I'm Sarit. Kathy is here too, and we're going to be talking about RNA and cancer, more than just the messenger. This is a very challenging topic, and I'm really glad to be here with Kathy, who has just a wealth of knowledge about this area. So I'll just be the doctor sidekick to her brilliant science knowledge. Anyway, so why should we pay attention to RNA and leukemia? This will be the overview of our presentation. This is what we're going to talk about. We're going to compare and put in context the differences between DNA and RNA. We're going to talk about what are some of the things we know go wrong with RNA, specifically in leukemia. We know quite a bit about DNA. RNA is a nascent field that is full of potential. We're going to look at what research is being done to target those issues in leukemia and what technology is available that is RNA based. Kathy, I'll let you explain the timeline.
For sure, and I want to just say that I'm really so happy to be giving this talk with such an outstanding physician and the chief of hematology at the Jewish. So you are no one's sidekick. In terms of why I think about RNA research, well, this is just a bit of a timeline. Hopefully you can see my cursor, kind of showing you that from the discovery of messenger RNA back in 1960 to say COVID, where all of us have probably been injected with an RNA-based medicine in the COVID vaccine from Moderna and Pfizer, that in a mere 50 years, things have really accelerated. Having said that, just as Sarit said, I think there's a lot more to know, but I think for sure now we can really start to imagine using vaccine technologies or other technologies we'll discuss today, eventually in a more sort of therapeutic setting. So as we just want to first start off and explain the differences between DNA and RNA to you in order to give you sort of a comparator, because we know DNA and the double helix and Watson and Crick and all these names are very prominent in popular culture. So just to kind of give you a comparison, we're going to talk about RNA relative to DNA because they are very similar. And this is just showing you what a normal cell looks like. And we think about DNA, or deoxyribonucleic acid, being within the cell nucleus, where one of its main jobs is to be the blueprint or plan for each given cell and eventually the entire organism. And if we think about it, it's interesting as well that we have trillions of cells in each of us. And so the amount of DNA and RNA we have in each of us is really an amazing astronomical size number. But in each of our cells, we have the blueprints to remake us in essence. And this is important, but this blueprint can be altered, as we'll show you as we go along, by modifications to the message of DNA, also called RNA, which goes to the cytoplasm and instructs from the blueprints for the cell how to build, say, a blueprint for the house, and then in the cytoplasmic side, the RNA allows that house to be built, if we can think about it like that. I don't know if you want to say a few words about DNA, Sarit?
Right, so DNA, so we think of DNA as the blueprint of the cell, meaning that it contains all the information about what the cell could potentially do, right? Could it instructs the cell whether it should divide or not. sorry, it has information about whether the cell should divide, how it should divide, what functions it should have within the cell. But and so that's all contained in the DNA, which you can see it is in the nucleus. The nucleus has these chromosomes, which are tightly wound DNA around histones. And then as you see it open up here, you can see that actually DNA has an alphabet code made of 4 nucleotides, guanine, cytosine, adenine, and thymine. And so that's the code. So the code is all these nucleotides associated in different orders, and the orders define the gene, and then the link or the chain of the chromosome of the DNA is wound in the histone, and then the histone wounds into a chromosome. So it's nice and tightly packed within the nucleus. And that is what's called the genome. And then when something needs to happen, so the blueprint needs to be brought into effect, or the house built, or the element of the cell function has to occur, the DNA gets opened up by RNA polymerase, and then an RNA transcript is, gets opened up, and then the RNA polymerase will make the RNA transcript, or the mRNA, or messenger RNA. And this messenger RNA is different from the DNA. It has different nucleic acids. Well, 3 are the same, adenine, guanine, and cytosine, but then uracil is a different one. So that's one way in which we distinguish them. And the other thing with RNA is it's much less stable than DNA. It will break down eventually over time once the transcript has been used. And we're going to get into more detail about what happens to mRNA. But ultimately, the DNA gets opened up and transcribed into RNA, and then the RNA will make the proteins that allows the cell to execute its functions. And so this is just to show you the difference between DNA and RNA. On the right, you can see that the DNA is a tightly wound double helix with these nucleic acids. On the left, you have the RNA, which is a single strand that is a mirror copy of the DNA, but instead of having the T, it has the uracil, right? The other difference is the sugar backbone, which is different between the two structures, so DNA and RNA. And that you can see over here. So as the title says, the sugary backbone, one atom makes all the difference. So on the left, you have the DNA that has the hydrogen, which allows twisting into the helix. And on the right, you have the RNA, which has the oxygen and hydrogen that allows for chemical reactions and allows for more diversity in structure. It allows the RNA to be read and manipulated more within the cell, which Kathy can explain in greater detail. Okay, so
I'll be happy to later.
Later, right. Okay, so the simple things are the errors in DNA. Those are fairly straightforward, at least for me. And here you have one error, which is called a point mutation. You can see there's like that little blast of orange and red showing that there's a mutation that occurs in the DNA, either from radiation therapy or exposure to chemotherapy, and often for reasons we don't know. And then as the cell divides, the DNA will also replicate itself with the same error. And that's an error in the blueprint, which means that when the RNA is transcribed, then there will be an error in the RNA. And then when you make the final component, which is proteins, or here the analogy is to a house, the house will have too many fireplaces. So it will be an abnormal house. So and that abnormality could be, for example, translated into a protein that's abnormally produced in a cell. Or, yeah, next slide. Okay, so that's a point mutation. Other errors in DNA that occur are errors where the DNA chain is broken, or 2 chains are broken, and then they reassociate. And on the bottom left, you see a whole new genetic code made for a protein. And that is a good example of that is CML, where you have the BCR-ABL rearrangement. So there's a chromosome 9 and 22 rearrangement that results in this BCR-ABL gene. And that gene eventually becomes an mRNA, and the mRNA becomes an abnormal protein. And in the case of CML, it means that you make a lot of white cells, and so many houses, and some of them are also abnormal in their appearance. Then on the upper right side, you see that there can be breakages in the chromosome without a rearrangement, and then the whole part of that chromosome is lost, goes into the trash. as you can see from the figure, and the protein that used to be made, often proteins that control cell growth, is lost, and then the cell can just grow in an uncontrolled manner. So those are the common things that we see: point mutations, breakages that result in loss of chromosomes, or chromosomal rearrangements. We also see duplications that make more protein. Those are DNA errors, and they're common in both leukemia and lymphoma. So then we come to RNA, and I'll pass it over to Kathy now.
So obviously the DNA world is incredibly important, and I think one of the reasons that DNA is the blueprint is, as Sarit pointed out, it has an increased stability over RNA, which makes it a more stable blueprint over the many times cells have to divide, or cells have to turn into different types of cells, like different types of blood cells or neurons or whatever. But as we sort of alluded to, that one of the ways that DNA communicates with the cytoplasm in order to make, say, the ultimate actor in a protein is by sending this message, which is called messenger RNA. And so it turns out, though, that only about 1% of all of the RNAs made in the cell are actually making these messages. And so for many years, this was really the focus, is understanding the messages. And it's very important because, as you saw, we could have houses with 3 chimneys everywhere causing havoc. But it also, all of these other RNAs we call non-coding, because they don't make a protein, are playing critical roles, and they can also be disrupted in cancer. And so there's sort of, if we think about the different types of RNAs that we can talk about, we have RNAs in the nucleus that are involved in making the machines that make proteins. So these make ribosomal RNAs, for instance, which we'll talk about a little bit. We also have RNAs like the messenger RNA and other components of the protein synthesis machinery, such as tRNA. So this is, for instance, of what a messenger RNA looks like, and this is what a transfer RNA looks like that's important to translation. And you can see, simply because of this extra oxygen on the sugar that Sarit pointed out earlier on, this one atom change allows the RNA to fold into much more complicated, intricate shapes, some of which you can adopt for our own purposes and bioengineer into things such as aptamers that Sarit will discuss later as bioactive drugs that can be used. And others are very important for the functionality of the molecule. And so if the structures get disrupted, then you can have all sorts of problems in the cells, which can cause them to grow or not to differentiate, et cetera, that we see in cancer. So the different types of RNAs vary in the sequence, and the sequence is the A, the G, the U, and the C that we talked about before. Those 4 nucleotides are our code, or our blueprint from the DNA, which gets transferred in a mirror image in the RNA when it's a messenger RNA, or is important for how these other molecules actually function. So there's many types of RNAs, and this is a whole new world of biology which people are trying to understand and then to leverage to develop new treatments eventually. But there's a lot to know. And the other, I think, important thing to understand as we move forward is that some of these folded RNAs actually have the ability to promote chemical reactions in the cell. And so they have this, very active chemistry that allows them to both be important for their functions in normal physiology and to be dysregulated in leukemia and lymphoma. So if we think about sort of, it's a little bit like the diagram that Sarit showed you earlier, in that we have the DNA here is in blue, and in red is an RNA message, which is sort of a mirror copy. So this copy can be edited inside the cell as well. And so we can start to have changes to the RNA that aren't related to changes to the DNA. So we can have changes to the RNA that don't need to have a chromosomal translocation like BCR-ABL, and they don't need to have a mutation in the DNA. So when things go awry, they can go awry, but the DNA template is untouched. And I think that's a really important concept, is that we can have this sort of rewriting of the message without touching our blueprint. And so in these cases, RNAs can be cut up and religated into different patterns, and I'll show you a picture of that in a minute. And once these RNAs are sort of ready for delivery to leave the nucleus and go to the cytoplasm, they go to this sort of powerhouse picture here, where they can be synthesized in proteins that we know can then have odd properties that can support like the cells being more oncogenic. And this is a sort of the same kind of idea of some of the steps that RNAs go through before they can reach the cytoplasm, and so how they can be reorganized without having to reorganize the DNA. And so I won't go into detail with the picture, but we can just sort of highlight a couple of steps that have both been shown to be disrupted in leukemia and lymphoma, and actually maybe actionable targets. Whether we have the best drugs to target them yet or not is unknown, but at least knowing what to target is obviously an important step. And so, for instance, in AMLs and in MDS, there's a series of mutations in the machinery that causes the cutting and restructuring of these RNAs, which contributes to the poor prognosis of many patients. And we don't have to have mutations. We can have overexpression, we can have the inappropriate elevation of other factors that can also change splicing, but in this case, without changing the DNA. So we can have two levels where we can have this process sort of hijacked by cancer cells. And other processes include the exit of the RNA to the cytoplasm turns out to be highly controlled. And so if you want to, for instance, if a cancer cell wants to be able to promote its cancer state, it will perhaps increase the export of the RNAs that encode proteins that will help the cell invade other tissues, help the cell make more cells, help the cell escape stress and death. One of the other things is that the RNAs, as we talked about, I've sort of alluded to several times in this last few minutes, is that some of these RNAs, that 1% of RNAs that are the blueprints for making protein, get converted from the RNA into the protein in a process that's called either protein synthesis or translation, because it's translating the genetic code into protein. So that's where this word comes from. And even this process can be dysregulated in cancers, and people are trying to understand better how we can have modifications to the machinery that does translation in order to elicit oncogenic effects. And so one of the things that, for instance, can be done is that in the cell we have a ribosome, and that's our reader of the RNA and our maker of the protein. And the enzymatic or chemical reactive part of this molecule to make the protein is actually the RNA. And these RNAs, called ribosomal RNAs, are very evolutionarily old. They're from even found and highly similar to those that are found in, say, yeast. So this is a very old chemistry is what I'm trying to say. And I guess it's because it works well. But if you have, say this is our RNA code and we can see all of our little nucleotides, all we have to do is do something called a frame shift, where if we have a sentence and we're reading a code and the sentence says the sky had sun, but if we just make our three letter code off by 1, all of a sudden we say the THSK adds on, right? It makes no sense, right? And so the ribosome in cancer can have this kind of effect. And if you can see that you either completely make a different protein than you wanted, or you may end up not making anything sensible at all. And so these can also lead to sort of these kind of cancer the cancer causing potential of a cell. The other thing that can happen is the ribosome machinery can just stall. It just gets stuck, and it can't have the little, like, almost ticker tape of RNA go through it any longer. And that causes proteins to be made that aren't full length, and those can end up being thrown away, sort of like when we talked about those bits of DNA that were lost. They'll end up in the trash, as Sarit said. So that can happen. And if you had an important protein that was stopping the cell from growing too much, and that happens, then obviously you have another potential way that this sort of RNA biology is contributing to making cells cancerous. There's what's kind of interesting as well, from the sort of RNA nerd in me, is that to make protein, it takes DNA, because DNA was our blueprint, if you remember, to make the house. It takes 3 different types of RNA in order to make protein. So a lot of us are very RNA based, is I guess what I'm trying to say. So maybe, Sarit, you want to take over for a minute, or? You're on mute.
Yeah, sorry about that. So just to recap, then, in cancer, we have, well, firstly, DNA gets transcribed into RNA, RNA gets transcribed into protein, and the RNA to protein step is an extremely complicated step that is highly regulated, as Kathy said, and in part regulated by other RNA molecules, but also regulated by proteins. And so it's a really complicated kind of structure. And that's what this is trying to say here, that RNA binding proteins regulate RNAs, and RNAs regulate production of proteins. So it's a bit of a, so you could imagine, let's say, an mRNA that has proteins that control it, but mRNAs also make those proteins that eventually will control it. And so it's a very complex system that is well balanced in a healthy cell, but can become disbalanced at different stages in an unhealthy cell. And we're going to show you some therapeutic examples of that. And that's basically what this slide is saying. So what do we do? What do we do about RNA disruptions? Or how do we leverage the role of RNA in the cell to treat cancer? So depending on the issue identified, knowledge of the RNA machinery can allow us to target faulty portions of RNA machinery with conventional drugs, like chemical inhibitors, biological products like proteins, and you can use RNA as the tool itself to fix the problem. So technology exists to synthesize and package RNAs and inject them into the body. And the COVID vaccine is an example of using mRNA as a tool, a therapeutic tool. All right, so let's talk about some of the drugs that have been developed or are being tested in that pertain to RNA in leukemia in particular, and in some cases here we're going to be talking also about lymphoma and other cancers. So we thought we'd focus firstly on the RNA splicing mechanism. So as Kathy said, the RNA is produced, and then there are enzymes that will control its length. So you can see up on the top that some of them get cut shorter, and then or the full length is kept. And the length of the RNA will determine the protein product, whether one is made, which type of protein is made. And yeah, you can see that on the bottom right. On the right, there's a shorter protein, and on the left, there's a longer one. I'm not sure, Kathy, do you want to explain that in a little bit more detail?
Sure. So when a messenger RNA is made, it has parts that will become protein and parts that make it sort of like an IKEA modular furniture, where we can just mix and match these different colors to make proteins with different functions. And partly that's a normal process of the cell in order to adapt to various cellular stresses. But if it goes wrong, and you have, for instance, a mutation or dysregulation of factors involved in the choice of which protein, you can end up having, say, an oncogenic protein or a dead protein, as it were.
Right. So, okay, so in acute leukemia and in myelodysplasia, there are, we said this earlier, that frequently we find mutations in splicing factors, and those 4 genes in particular, which you may have heard of, particularly in the context of MDS and also in acute myeloid leukemia. And in the next slide, we'll talk about targeting those genes. Okay, so here it is. Okay, so you can see here, for example, there's a gene called IRAK4, and this is the mRNA for IRAK4. And if IRAK4 is properly transcribed, you form the short IRAK4 mRNA, which is rapidly targeted for degradation. And then, however, when you have those splicing mutations that I listed over there, you can produce what's called a long IRAK4 mRNA because the splicing does not occur. Those little loops of RNA are not cut out, or the golden part. And so you get this longer mRNA, which produces a longer protein. And the longer protein does not get degraded, and rather works as an activation of signals leading to cell survival and proliferation. It's also involved in immune response. And so there have been attempts to try to target the specific splicing mutations, which have failed. So now the approach is to look at this drug called emavusertib, which really blocks IRAK4 long and can result in clinical responses in both lymphoma, myelodysplasia, and acute myeloid leukemia. In acute myeloid leukemia, it also targets FLT3, but I think because of both the IRAK4 and the FLT3 inhibitory function, it has maybe some beneficial effects beyond just FLT3 targeting. So this is sort of an indirect way of addressing a problem with a splicing abnormality. Right, so that's what you see here, that this H3B 8800 drug, which is a splicing modulator and binds to SF3B1, worked in animal models, but was not efficacious in patients with these diseases. However, when you kind of approach the problem indirectly, at least early data suggests that this might be a productive and a good approach. And we're actually doing, Kathy, a clinical trial of this drug, emavusertib, with in patients with low-risk MDS to see if you can improve their dependence on transfusion dependence.
Does it look good, or
Just-
too early to say?
We're just starting.
Okay.
Yeah.
Good luck.
Thank you. I'll give this one to you.
Okay, so another sort of emerging drug of interest is selexinor, and it targets, if you remember, I mentioned the trafficking of an RNA or protein out of the nucleus to the cytoplasm allows a way for the cell to control its cytoplasmic availability. So if the RNA never makes it to the cytoplasm, it cannot be made into a protein. So it's a way to reduce proteins without altering the DNA at all. And so this step is called nuclear export, and a group called Karyopharm found this drug here, selexinor, was able to stop the export of certain proteins, and particularly at the time they were focused on a protein called P53, which is a really famous protein that drives a great many cancers. But it turns out that it also influences the export of RNAs as well. And it seems to be that perhaps some of its therapeutic benefit is arising from this RNA export function as well. And so this is in a case where we have like normal export. We can see that our little factor is a little carrier protein, binds a cargo and allows this P53 or RNA cargo to arrive to the cytoplasm. And when selexinor is added, it kind of stops our little carrier protein from being able to traffic the cargoes into the cytoplasm, and thus is a very strong inhibitor of the RNA export, and for some RNAs and for some proteins is a way to sort of slow down cell growth and reduce survival of cells under stress and things like that. So I think this one is for you, Sarit.
Yeah, so right, so selinexor then is approved for the treatment of large cell lymphoma by the FDA, but not approved in Canada. But in Canada, we do have access to selinexor for the treatment of multiple myeloma in combination with bortezomib, I think, and steroids. And so this is a proof of a sort of RNA-based approach to treatment of these blood cancers. I think in leukemia, the results were not as favorable. I believe that it was tested in an early phase, but did not show enough efficacy. But this is a drug that's kind of a little bit non-specific, you could say, and it is preventing the movement of abnormal proteins, but also normal proteins. And so patients tend to have quite a few side effects from this: gastrointestinal, diarrhea, weight loss, nausea and vomiting, low blood counts. So it works. It works, but it has some side effects.
And I guess, Sarit, it also kind of speaks to the fact that we're looking at some of these drugs that we just mentioned, the splicing inhibitor, the IRAK4 and Selexinor, in the context often in the lab and early trials as single agents. And I guess one of the questions that has to really be addressed is, you know, what are the things best to combine with some of these to see if we can really leverage them.
Yes, it's very true that for most cancer therapies, you know, giving these drugs alone is not going to result in meaningful responses. But as we'll see later with some of the immunotherapies, that may not be the case, but that's for later.
Do you want me to take this one, or do you?
Sure. Yeah.
Okay. So as we sort of said, Selexinor is involved in blocking the export of both proteins and RNAs out of the nucleus. And so one of the pathways for export it uses is something called EIF4E, which, full disclosure, is something that my lab is very interested in studying. And through a unique mechanism shown here, which I don't think is really important, but I'm happy to answer questions in the question time. But what I really wanted to just show you is that some RNA binding proteins, such as EIF4E, this factor, they can be highly dysregulated in AML. Sarit and I have done Sarit's done the clinical trials, and we've done the correlative studies to show that targeting RNA factors like 4E can lead to clinical benefit. But also that some of these factors can be involved in many of the steps. So they can really kind of rewrite the ultimate RNA and protein composition of the cell without actually changing the DNA, which is sort of an interesting way to look at how leukemia and lymphoma drivers work versus the sort of traditional DNA mutation, et cetera, way, which is obviously very important too. So that's really what I wanted to show you, is that some of these factors can act in many different steps. And so dysregulating one factor doesn't necessarily dysregulate one step, it can dysregulate many steps, causing a cascade of downstream problems. So Sarit?
Well, actually, so Kathy and I, like I said, or like she said, worked on many clinical trials of ribavirin, 3 of them in total, in patients with acute myeloid leukemia. But before I get to that, I wanted to ask you, Kathy, how you determined that we should be testing ribavirin as an inhibitor of EIF4E in acute myeloid leukemia and not other cancers. I should know, and I think I remember, but it'd be nice for the audience maybe to hear.
Well, it's partly historical. So my lab had been working on PML, the promyelocytic leukemia protein. And so some of you in the audience may have heard of acute promyelocytic leukemia, APL, which is now quite treatable with retinoic acid and arsenic. And Sarit can tell you way more about that. But at the time I had developed an interest in that area. And so my lab just tested panels of other AMLs. We didn't see 4E dysregulated in APL, but we saw it dysregulated in many other leukemias. And so that kind of sparked our interest, since we were sort of already in that space, not to you know, other people have tried in solid tumor disease. I think, yeah, they're even listed here: prostate, follicular lymphoma, and head and neck. But the reason we started with this was really historically rooted to a serendipitous observations with the promyelocytic leukemia protein PML. So I don't know if that helps, Sarit.
Yeah, thanks. I think you told me this way back in 2006 or something.
Yes, yes, we have collaborated a long time, which is awesome.
So given that we went, we had a clinical trial, firstly of ribavirin alone in patients with acute myeloid leukemia, in whom we could show a high level of EIF4E, because it's not all acute leukemia, myeloid leukemias that have that, it's about 30%. And so if the patient had a high EIF4E that we measure, that Kathy would measure in the lab, then they would be eligible for the trial. So the first trial was really just the ribavirin alone. And you can see in the panel on the right there, there's the patient before treatment, and you can see there's a whole lot of leukemia cells. And if you look at the green cells in the black background, they had a lot of 4E, EIF4E, and it was in the nucleus, so a lot of green stuff. And then this one patient achieved a complete response, meaning all the leukemia cells disappeared and the blood counts normalized in the peripheral blood. And if you look at their remaining few cell blasts, they have no high 4E or very, very little. So they're really in appearance or in phenotype completely different from what was seen before the treatment. So that was a proof of concept that giving ribavirin to target EIF4E can result in remission of the acute leukemia. So that was really quite exciting. And then we did other trials where we combined it with chemotherapy and also saw some meaningful clinical responses. And now we're looking to consider giving ribavirin in combination with the more standard treatments for acute myeloid leukemia, currently including venetoclax and azacitidine. So that's hopefully something we'll do in the not so distant future. And I'll let you explain. Oh, okay. I don't know if you want to explain what's going on molecularly.
I think maybe we should keep going because of time. And if anyone wants to ask me, we can go back.
Okay. Okay. So that's how we at the present time, I suppose, target RNA. And as you can see, a lot of it's not direct targeting, it's more indirect through ways of controlling how the RNA is able to carry out its roles and responsibilities within the cell, many of which in cancer are bad for the organism. So, but this is using RNA as a medicine in itself. So I'll give you some examples. Right now, there aren't really any in acute leukemia, certainly not in the clinic. And so these are just categories of RNA therapeutics. There's RNA aptamers, which bind a cell receptor. Then you have antisense oligonucleotides, which will, or siRNAs and miRNAs, which will bind to the mRNA itself and prevent it from being translated or reduce its stability, or again, or cause it to degrade. So different ways of making the RNA not function. And then finally, you actually can use the mRNA as the therapeutic itself, and that's the concept of the mRNA COVID vaccines, where you generate an mRNA strand that will eventually enter the cell and use the person's cell to make the protein. And in the case of the RNA, that protein is immunogenic, meaning that your immune response will recognize it and build an immune response or resistance to the larger target, which in the case of the vaccine was the COVID-19 virus, but in cancer can be a cancer protein. Okay, so this is the example of the aptamer. We don't have any of these in the cancer world, but there is the one that's used, and I just learned this for purposes of this talk.
I'm sorry.
That's okay. So it's the VEGF. It inhibits binding of VEGF. Sorry, it binds to VEGF and inhibits its function, and it's used in macular degeneration. So it's like a small protein, and it's injected right into the eye. So what are the advantages? It's a smaller molecule, has the potential to get to hard to reach spots, can be taken up by the cell, and is easy to produce in larger quantities in comparison, say, to an antibody. and it's less likely to be immunogenic for the body. So that's one, a successful one in macular degeneration. There is one apparently being developed in AML with some encouraging results with more work to be done. I haven't seen it myself in the clinic, but there's a lot going on in AML.
I think it's still in the animal model stage, so yeah.
Oh, okay. Do you want to talk about this one?
No, I thought this was for you, but I can if you want.
Okay. Okay, so this is an antisense. I remember like 15 years ago, 20 years ago, we were using antisense to BCL2, which is a anti-apoptotic protein, and they didn't work. They didn't work because I think they were not potent enough, but also they caused some liver toxicity, so they were abandoned. But now I think people have continued to look at antisense onto oligonucleotides, and as I said, these are small fragments of RNA that bind to mRNA and prevent the protein that they're targeting from being translated. So being not the protein, but the mRNA to be translated into the protein. So they bind here, as you can see in the diagram, and then you get this red light, meaning no protein made. And there is one now to GrabB2. GrabB2 is involved in the signal transduction from tyrosine kinase signaling through the cell, and the early data suggests that it actually can be given with chemotherapy safely, but whether it's actually effective has yet to be determined. Okay, this is a similar idea of targeting this BCL2. I'm sure many of you have heard of BCL2 because you probably are familiar with venetoclax. So venetoclax is an oral small molecule inhibitor of BCL2, which is highly effective in CLL and in acute myeloid leukemia. And so this is an siRNA, again, same idea as the antisense, which is to block the mRNA for BCL2, have it degrade, and then you don't make the BCL2 protein. And it may be better, perhaps, or complementary to venetoclax because there is some resistance to venetoclax, and targeting the entire protein, and the resistance to venetoclax is through mutations in BCL2, but this siRNA could probably just not care about those mutations and still break down the mRNA. So could be a more effective therapy, or one that can be used once people become resistant to venetoclax-based treatments, and that's in phase 1 testing at this time. Okay, so then other potential targets are miRNAs in acute myeloid leukemia. Those, haven't seen those yet in the clinic, but I'm sure that they're going to appear soon. The ways to target them are with antagomeres to the miRNA 21 and 196B, which have shown some activity in mice. The same idea, they don't allow the miRNA to function. And then there's an miRNA 181A, which plays a tumor suppressor role, downregulated leukemia and in CML. And so the question is, would it be beneficial to increase its levels? So in some cases, try to eliminate the mRNA, in other cases, try to enhance its function. Okay, and these are just a list of clinical trials looking at targeting miRNAs, and based on the slide, you can see that some of them are in hematological malignancies, mostly in lymphoma and multiple myeloma, less so in acute leukemia. Okay, so here, well, in lymphoma in particular, immunotherapies, and in solid tumors, immunotherapies are really hot topic. In acute myeloid leukemia, immunotherapies are a little more complicated, particularly the CAR T-cell technology. So, and the CAR T-cells, but also the checkpoint inhibitors. Checkpoint inhibitors are drugs which allow the immune system to be turned back on when it's been shut off sort of by your cancer cells. So those checkpoint inhibitors and CAR T cells haven't worked very well in acute myeloid leukemia, but in some lymphomas, they're quite good. Anyway, the way a CAR T cell works is that you have a adenoviral vector, so viral vector with a part of the B cell CD19 gene attached to it that is transfected into the T cells, and then the T cells will express the CD19 on its cell surface. or anti-CD19 itself surface, and then when it comes in contact with the malignant cancer B cell, it will turn on its functions, kill the cell, recruit the immune system to kill the lymphoma cells. And so the viral vector can be replaced by an mRNA vector. I'm not sure, Kathy, do you think it's a more efficient way, or do you think it's just a safer way? Why do you think people are looking at using mRNA rather than sticking to the, I don't know, or viral vectors to transfer cells?
I guess perhaps to alleviate concerns of people receiving viral vectors, because there is sort of, I think that everything is being FDA approved or Health Canada approved and is safe, so I don't want to say that it's not, but I think it overcomes maybe a conceptual bias that people are a bit nervous about it. And I also think that it could be that once you develop the system, then it's very easy now that people have these nanoparticles to be able to just reinsert different RNAs in order to modulate functionalities. So isn't it true that in some of the CAR T people that they actually lose some of the CD19? And so maybe there's ways that you could do a second round, but with some variation in this in order to represent another antigen that could be recognized. I'm not sure. That's speculation.
Yeah, no, I think you're right. I think there's a question of efficiency and a question of safety. And the other thing we're showing here is the actual concept of mRNA vaccines for antigens that are seen in tumor cells, like in melanoma, and KRAS for solid tumors. And that could conceptually eventually come to AML, where we have some of these, we have RAS mutations and we don't know how to target them. But I think in the leukemia world, in particular in AML, the immune environment is not necessarily one that lends itself so well to immunotherapies, at least not in 2024. So that there's a challenge there in adopting these treatments for acute myeloid leukemia and MDS. But no today doesn't mean no tomorrow. It's just a matter of how to do it.
Well, if you think about how far since 1960, and it's almost, you know, it's a long time ago, and how far we've come, it'll be really interesting to see like what if we could have a time machine in like 50, 100 years to know what people will be doing with all of this.
Right.
But I guess here it also says there's some early examples in cancer trials. I don't know if you wanted to mention those or.
Yeah, so these are the things I was saying. And so the mRNA type vaccines, you see that, well, the first one codes for immune boosters such as OX40 ligand, IL-23 and IL-36. So those are going to, you know, further enhance the immunological response that you can get from checkpoint inhibitors. So my guess is that these will eventually be combined with checkpoint inhibitors to make them better in solid tumors and in lymphoma, Hodgkin's probably. And then there's personalized mRNA vaccines in melanoma. I wonder how that would apply. So meaning that you find antigens that are expressed by melanoma cells in a given patient. And I think, well, maybe correct me if I'm wrong, but mRNA technology is pretty scalable or versatile or easy.
Adaptable.
Adaptable, yeah.
Yeah.
So which is why you can have personalized mRNA vaccines. I mean, you have CAR T cell is more or less personalized, but it is a big challenge to produce. I wonder if this is easier. And then the last one is oncogene mRNA vaccines such as KRAS and solid tumors. KRAS is it's a problem in solid tumors. It turns out it's a big problem in acute myeloid leukemia too, KRAS and NRAS mutations, and they're not that targetable, at least with the drugs that well, certainly they're actually not at all targetable in leukemia, but you know, if this could work in AML with KRAS mutation, that would be quite nice, quite a breakthrough. Oh, okay, so we talked about this when the mRNA joins in for the CAR T ride. So I think you know how CAR T works. To make CAR T cells, human T cells are isolated and engineered in the lab to express a receptor that will allow T cells to recognize and kill cancer cells. And this requires modification of the T cell DNA, which is often done with viruses. And as I said, now new technologies are being explored to introduce DNA without the use of viral vectors, and the DNA is introduced to T cells along with the mRNA for the enzyme needed for the integration of the sequence into the T cells. So I don't fully understand that, but I do know that it's a way to avoid using a viral vector. Okay, so that's it. That's our talk. I mean, Kathy, would you like to list the conclusions here?
Sure. I hope that today you've come through understanding the relevance of dissecting RNA biology. It's a long-term investment for sure in terms of like how often, well, how frequently we'll see it in the clinic. I think it's just going to burgeon, but without understanding the basic scientific underpinnings of what's going on in the cell and why RNA is different than protein and all of the different things that RNA can do, that we can't really leverage that information for developing drugs either to the proteins or not. So having that scientific basis, I think, is critical for us to build a solid foundation to develop new therapeutics. And that's sort of why Sarit and I went through sort of showing you the complexity and elegance of RNA biology. And I think that it also allows us opportunities to target things that were previously considered untargetable because now are undruggable. I think now it allows us to drug things like transcription factors, et cetera, which were considered too hard to drug before. I don't know if you have anything you want to add to that, Sarit?
No, that's, I mean, I agree. I think that knowing the biology of the normal cell and how it's disrupted in malignancy is really the foundation for discovering new treatments, and takes a long time. But as you can see, the pace of discovery at one point sort of just takes off once we understand, you know, what the function of a particular cellular process is. So yeah, more work and more hard work, like from people like Kathy.
And from people like Sarit. So I guess, well, thanks to LLSC for giving us the opportunity, and for the audience for listening, and for Sarit, all your wisdom, and a special thanks to Carolyn Lambert, who helped us very much with preparing for today.
Yeah, thank you, Caroline.
So I think over to you, Paul.
Yeah, okay. Well, thank you so much to both of you. This is a very, very complex topic, and you did a wonderful job explaining this. And we do have a number of questions that have come in relation to what you've talked about. So it is time for our Q&A period. If you haven't done so already, if you have any questions, please put them into the Q&A box in the webinar panel. As we mentioned earlier, we may not be able to get to all of your questions, but we promise to answer as many as we can. And note that there will be no specific questions asked regarding health history or status. So, as I always do with these questions, I like to start specific, and then we'll get broader, looking at the overall message and the hope that we're trying to achieve with the work that we're presenting here today. So, I'll start with the very first question: is simply, what is the Philadelphia chromosome? I know we touched upon CML and you know, these chromosomal translocations that can happen, but perhaps you can succinctly explain.
Yeah, so the Philadelphia chromosome refers to an abnormal rearrangement of chromosome 9 and 22 in the cell, and the product of that rearrangement is a gene called BCR-ABL1, and BCR-ABL1 will make the BCR-ABL protein, which drives chronic myelogenous leukemia. So if you don't have the Philadelphia chromosome, you don't have BCR-ABL1, you don't have chronic myelogenous leukemia. So that it is the defining genetic abnormality in that disease.
Wonderful, thank you. We have, does mRNA splicing editing or /editing happen in normal cells as a normal process, or is this something that is specific to, you know, cancerous abnormal cells?
I can take that one. It's something that happens in every cell, and it needs to happen, but it needs to happen at the right place and the right time. And so in order to produce appropriate proteins. So you can't just kill it because you need it to happen. So you have to be more nuanced about how to attack the cancer cells only, or the cancerous event only.
Thank you. We have, has selinexor been trialed in children with AML?
I leave that to you, Sarit. I'm not sure.
Yeah, I'm sorry, I actually do not know. But you could look it up on clinicaltrials.gov and see if that's been done or is in the process of being tested.
Right, and that's actually a great leadway into or another question is about where is the best place to find clinical trials that would be, you know, on this type of work, and how does someone, you know, get the information on this?
Yeah, can I just add something about clinicaltrials.gov? I think is really important is although it's sponsored by the US government, all around the world, if people want to publish their trials in journals, they have to have gone and put their data on clinicaltrials.gov. So you get an international view of the trials. I don't know, there could be, if Sarit, if you have an additional resource, but I think
No, that is the best. There are like provincial, I think, database or listings of clinical trials. Even some larger institutions will have those on their websites, but the best place is clinicaltrials.gov. It's quite user friendly, and if you identify a trial, you can see if it's open in your city. Yeah.
Perfect. And we have one question on what is your view on the use of mRNA from PCR blood from blood tests for early detection of cancers?
I think it's a really cool emerging area, and now that you say it, I realize that there was a part of a slide that I should have said something about, which was that there's also RNAs in these little little spheroid structures called extracellular vesicles that are in all of our plasma. And so there's been a lot of hope that this could be developed as sort of tests, you know, maybe every year as part of your annual checkup, they could just do a blood test and see if you have cancer X, Y, or Z. But I don't think, generally speaking, although Sarit can correct me, that it's up to the stage where it would be used routinely in the clinic, but certainly it's being used in trials.
Well, okay, so we have time for just a couple more questions, but so one, I'll try and bring a couple together here. So a lot of the work, you know, we're talking about here is leukemia and acute leukemias, and that are not very poor responsive or poor response to therapies generally. So how, for those cancers that don't respond to therapies very well, such as AML, will an approach using this RNA technology be more effective than what was currently available for, you know, with standard treatments?
Kathy, would you like me to try that?
I think you could. I mean, you're the clinician here, so you can start.
Okay, acute myeloid leukemia is a complex disease, and it is a grouping of diseases that right now we group based on mutations at the DNA level. So the early stuff that I talked about. But as Kathy's work, and then so that's standard. And then we can really kind of select therapies based on those very, I would like to say, rudimentary abnormalities, because the story is much more complex than that. And Kathy's and my work in targeting EIF4E in people who, you know, high EIF4E who have all these other mutations, but in fact, EIF4E is also a driver of their cancer, just speaks to how complicated the whole area is. So yes, I think that, you know, understanding more the molecular complexity of AML, including the role played by RNA and RNA, all those RNA structures that we discussed, will make a difference in terms of better selecting the patient for the therapy and the therapy for the patient. But I mean, it's a complicated road. Yeah.
Of course, yes, of course. So I think we'll end it on the last question here, just due to time, is that if you could have your ideal or dream study, what would it be? And this is, you know, no cost worries or anything like that. What do you think would be the best, you know, most effective approach to your research?
I let you go, Sarit. It's a clinical one.
Oh no, that's a Kathy question. You know, the challenge of clinical trials. Oh my God, there are so many, but one of them is all and they're needed, which is all the regulatory work that you have to do to make sure that you're, you know, safely administering the therapy to patient, and also then able to measure the outcomes. So we have a lot of great ideas and we want to do a lot of clinical trials, but the cost and the time and the overhead. So the ideal trial, and I don't just mean money, I mean like getting it right so that it's approved by the health authorities and that we get good results and everything. So the time and the expertise so the ideal trial is the trial that opens fast, that enters patients quickly, that you get results for quickly, so that you can, you know, quickly move on to the next thing or to make the right discovery. So there are so many questions we should ask, and it's more a question of how to efficiently ask them so that we get to our results in a more rapid way, so we can help more people more quickly. So I know I'm not answering your question, but that's my dream world where I could just do it quickly.
It's a very difficult question to answer, obviously, and I think a number of the questions that we're getting in here are, how does this apply to adults versus children? And we're not looking at it might be the same disease by name, but it's not the same disease by nature. So, can you touch upon how that how that plays into your approach as well for if we're in your work?
Well, you mean, oh, sorry, go ahead.
Well, I can take it and then you can follow, but for instance, when we worked in the lab with cell lines from ALL patients, which are pediatric, and also some of our AML cell lines are derived from pediatric patients. So the popular ones, like THP ones, Sarit, are from a very young child. So the cell lines that we could get, so we're not taking patient specimens, but cell line specimens, they can also display the same exact phenotype of having really high nuclear 4E. And if you treat them in the lab with ribavirin, you'll see the same changes. And other people have seen dysregulated RNA metabolism in pediatric, not necessarily trials, but in pediatric cell lines and specimens as well. So I think that it's not something that's going to be, because people's RNA biology is, it's from birth, right? You're going to need to do all of the things we talked about from birth. So, or even before, in fact. So I think that this is going to be something relevant to all ages. I mean, there'll probably be specific things that are more relevant to younger than older individuals, but the basis of the RNA biochemistry will still be relevant, I think. I don't know if you have anything to add there, Sarit.
No, I think that's pretty accurate. I think that things discovered in adults can also be tested in children. Often we start with adults because there are more of them who have cancer, and then bring our discoveries to children.
Wonderful. So I think we are reached our time here. So I want to say thank you so much to both of you for your time today. The expertise and your description of everything is wonderful, and I really appreciate you taking the time out of your busy schedules to take part in this for us in the beginning of our Leukemia Conference this week. So it's wonderful. Before we sign off, actually, Kath, can you switch to the next slide, please? And extra slide. So before we sign off today, I'd like to remind everyone of our upcoming conferences this week. AML tomorrow, followed by CLL and CML on Wednesday, acute lymphoblastic leukemia on Thursday, and then on Friday we have a conference on how to actively participate in your care. So please, all the details are on here, and please attend these events as well. Next slide, please. We'd also like to thank our sponsors. Thank you to AbbVie and Astellas and AstraZeneca, Bristol Myers Squibb, Jazz Pharmaceuticals, Johnson & Johnson, and Novartis for their support in making these conferences possible. Next slide, please. Please remember that we are here to help you. You can reach us by email, by visiting our website, bloodcancers.ca, or by calling us on the number here, toll free. Please note that a short survey will be sent to all participants after this presentation. Please take a moment to respond, as it helps us plan for future events. And last slide. Thank you all so much for being with us here today, and take care. Bye for now.
Thanks.
Thank you, everyone. Bye.